So in a similar format, if each of you could just take a moment to introduce yourself and talk a little bit about what you do.
Hello, I'm Asli Aykanat, and I completed my medical degree back in Turkey, and I'm right now in the Beggs lab working as a postdoc on zebrafish models with different genes like actin and rare genes like troponin, and we're trying to see if different mutations are pathogenic or the cause of the disease by using these animal models.
Hi everyone, I'm Matthias Lambert. I'm working with Lambert and also Lucan Carl at Boston Children's Hospital, basically on Duchenne muscular dystrophy, but also on Nemaline Myopathy for different reasons. And the first one is because I'm also living with Nemaline Myopathy. So I'm using zebrafish, I'm using also iPS cells, as you heard, and also some mouse models. So just to create models for rare population, just to understand the disease and then to treat the disease.
So hi all, I'm Alan Beggs. Some of you know me. I've been very focused on Nemaline myopathy for many years now, probably starting since about 1992. As Matthias just said, we also have another investigator in our lab, Lou Conkle, who he works with as well. And Lou was my mentor. Lou studied muscular dystrophy. And so from that, I kind of expanded my interest in Nemaline myopathy. And I'm PhD, not a medical doctor, but very interested in the translational applications of our laboratory research to the clinic. So kind of really making sure that what we do in the lab leads to benefit for the community as a whole.
Hi, I'm Jeff Widrick. I'm a muscle physiologist, and I look at these diseases as a problem in muscle function. And so as my colleagues understand the genetics and develop treatments, then I kind of step in and can evaluate whether these treatments actually improve the function of the muscle, so how much force or how much power the muscle can generate. And then the other side of that coin is there are many steps in muscle contraction, and different diseases affect different steps differently. And so you know, by working with these people, we may be able to reveal a step that we hadn't thought was affected, and it may illuminate a new path that could go down as a potential treatment. And I guess the last thing I'd like to say is my work is kind of disease or model agnostic, so it works for pretty much all the diseases that are represented here at this meeting. So I kind of cross over with a lot of the different researchers.
Wonderful. Thank you. Now, are there any questions from the audience?
Yeah, hello, my name is Chris. My son had a muscle biopsy done late of 2008 to figure out that he had Nemaline myopathy. As researchers across the globe, how important is getting new Nemaline myopathy patient tissue to hone in with the fish and the mice? Or do you already pretty much have the tissue you need to know moving forward to get better answers?
Hi, Chris, thanks. That's a great question, and actually, it's critically important. It's extremely useful. Actually, I could point to several of you in this room whose muscle has contributed to recent advances. There was a paper published last year that looked at the mechanisms of weakness. It's those muscle samples, by the way, that Jeff uses to measure the mechanisms of weakness. And so we never ask anybody to have a biopsy purely for research purposes. That may and does happen in the setting of a clinical trial. So if you're testing a new drug, then oftentimes a biopsy before drug treatment and after or during drug treatment may be done for research bases. We don't do that, but for those of you who have had a muscle biopsy done as part of your diagnostic workup, often there's muscle left in the hospital. For those of you who have enrolled in our group or who are registered with the CMDIR, chances are that muscle is either in a freezer in Boston in my lab or in Minneapolis in Dr. Lawler's lab. And in fact, Dr. Lawler trained in Boston, so we share things back and forth as needed. So it's actually critically useful and very helpful if you guys are willing to sign release forms that allow us to get that. It's not always available. The hospitals are very good about not releasing it if there's still a clinical need. So if they, in their judgment, think they may need to go back to that muscle in order to help manage your child's care, then they won't release it to us, or oftentimes they'll give us just a piece of it. So I think that the clinical needs are respected first and foremost. But maybe you can talk a little bit about what we do with these things.
Well, I just want to make the point that my work to measure the function of these muscle fibers, I only need a tiny, tiny, tiny little piece of that muscle. Yeah, if you gave me the whole muscle biopsy, I'd spend the rest of my career studying all those individual fibers. So a muscle biopsy, the point I guess I'm trying to make is a muscle biopsy can go a long way. It can help a lot of different investigators.
And I'll just say, I think you just heard from Dr. Lindquist a few minutes ago. A lot of the muscles they've studied come from you guys through my lab. And I don't know if the CMDAR registry's also contributed to you guys, but it's been really critical. And it doesn't matter who it comes through, we're all working together.
Alan, I just want to say something about that too. So with our mouse models that we study, the D286G mouse model, for example, was based off of a patient mutation. So anything that we can do to better understand the actual mutations, we can create those better mouse models. And that also helps us, again, figure out what mouse models match, quote unquote, what patient population. So anything that you can give us to help study and help you is great.
Oh, just a clarification. I know you meant to say Milwaukee, but you said Minneapolis, the Lawler Lab. Yeah. Just so everyone's not confused. It is Milwaukee.
East Coast, West Coast, sorry.
No, it's fine. That's fine, yeah. And just to continue what the discussion was about, the size of the muscle sample that's really useful. These are very small pieces of muscle, and a little bit goes a long way. I mean, we can take what's left over from a clinical biopsy and spread that out to a number of different researchers because it's just such a tiny amount is needed for what they are working on.
Switching fast and I can see the colors. Sorry.
Tell the colors.
My name is Johanna Dahl. I'm from Sweden, and my son Benjamin has NM. I tried to send some tissue to the CMDR. Yes, thank you. But since we live in Sweden and in Europe, we couldn't do that due to the GDPR law. Or they didn't want to accept it. Is that the same standpoint today, or I don't know if you could answer that, but because we want to contribute, but that could be hard.
So I don't know the specifics of that law, but I know there have been a number of new privacy regulations in Europe in particular that have made things a little more complicated. Maybe one of you guys would like to comment after me. What I could say is that there are also investigators in Europe working on Nemaline myopathy is very active, in fact. And so that might be one avenue for you to make it useful. The other thing is if you put the lab here directly in touch with the hospital or your doctors, sometimes we can work around things, do them a little more informally. If it's just a request, like for example from you to a hospital, they may say no, but if it can be transferred to a research lab and then a researcher sharing it after making it anonymous, taking your name off it, I would suggest maybe putting in touch whomever you were speaking with, so Stacy in this case probably, together with your physician And then maybe she can also, there's somebody in London who works on this. I don't know, maybe now with Brexit they're not going to be able to get your Swedish muscle. It's a little crazy, but we can push it. So, do you have any specific information about this?
I know that there has been a lot of changes, but I know that we still receive samples, and it's just about, it's a question of paperwork that changed a lot. And unfortunately, I don't have the details, but I'm sure that if you ask around, you'll find the right paperwork to sign, and hopefully the sample will be accepted.
More questions? Yes.
Yellow?
Go ahead. Okay, go ahead.
Okay, so my husband has a question I want to ask is, their cell models have been developed for CRISPR-Cas9 gene editing techniques?
So we are currently doing that in the lab. I mean, in my case, for tropomyosin, we are, just because, for example, there is a rare population, so again, we don't have sometimes access to the biopsy or to the cells from patients, and so we need to create the gene editing in stem cells, for example, and you can do cardiomyocytes or skeletal muscle or skin cells, what you want, just to study the cells and try treatment, because we have sometimes the technology, we have some drugs we want to test, but we don't have any models. In my case, this is, I mean, the reality. We have some drugs we want to test, but we don't have any models. So this is the opposite. Sometimes you don't have it. You have the model, you have the patients, but you don't have the drugs. And in my case, this is the opposite. So that's what I'm doing. I'm doing zebrafish, I'm doing iPS cells with CRISPR, and it takes time, but the technology now is advanced, and we know how to do that because we are doing that, for example, for Duchenne, and now for Nemaline Myopathy. I mean, it's working well. So I got already some cells, and I'm just studying now this year how the cells are working and how we can use these cells to do treatments. So yeah.
Sorry, there is one more. And the application of adenovirus system in a mouse model for gene therapy?
Yep, okay. So I think you read the word adenovirus, and I'm going to modify and clarify that what I'm going to talk about is adeno-associated virus, which is a different thing, similar name, and it's often referred to as AAV for short. Adenovirus is a very large virus that causes the common cold and has been tried for gene therapy a number of years ago, but actually led to a very bad outcome in one boy who unfortunately died. Adeno-associated virus, or AAV, is quite safe and is being used now to treat spinal muscular atrophy and is being developed to treat another condition, myotubular myopathy. And AAV doesn't cause sickness. The problem or the issue with AAV is that being a small virus, we can only fit a small amount of DNA inside. So if somebody has a mutation in a gene and they don't make that protein, we can potentially treat the disease by replacing the gene or providing extra copies of it that get carried in by the virus. If it's a small gene, there's no problem. We can fit it within the virus. The virus is basically just a little like bullet that carries the DNA into the cell, and then that gene will now make its protein and correct it. So the actin gene, for example, is small enough to fit within an adeno-associated virus. The nebulin gene, on the other hand, is extremely large. The protein that it makes is also very large, and so the nebulin gene is too large to fit inside any type of virus. There's a lot of effort and discussion and research going on these days to think about ways we might do this, and I'm involved in work to use AAV potentially to treat actin mutations, but not for nebulin mutations, and at this point not yet for any of the other genes, tropomyosin 3. How large is the TPM3 and the TPM2 gene?
They are very small, so basically you can use AAV if you don't have any expression of tropomyosin. You can use, I think, very easily AAV because this is a small gene. So basically this strategy, you can use it, yeah.
Although, on the other hand, I'll say that it also depends on the mechanism of the mutation. So using AAV brings in normal copies of something, and so if you're missing it, it will replace it. However, in the case of TPM3, you have one normal copy and one abnormal copy, and it's the presence of the abnormal copy that leads to the muscle weakness. So there, the challenge isn't to replace something that's missing. Instead, what we need to do is remove something that's there. That's also the challenge for actin. So the question then becomes, can we flood the cell with much more of the normal copies in order to minimize the effects of the abnormal copy? Or could we somehow turn off the abnormal copy?
Thank you. Are there any other questions?
Hi, my name's Verena. This is Xander. He's 10. He has Nemaline. Dr. Beggs, you just made a comment about working with the actin gene and the AAV because it's a short gene. And you also mentioned, and I know Xander has one functioning copy and one non-functioning copy. So the non-functioning copy is obviously created in Nemaline. So, I mean, what's holding things up? As a parent, obviously I can maximize his pulmonary, I can maximize his nutrition, but I'm forever looking for hacks. What can I do to actually help him right now? What can I do to make him a bit stronger? And I'm listening to, Dr. Gupta with the couch proteins. It's like, can I artificially stimulate an improved kind of couch protein in him? What can I do?
That's the critical question, I think, probably for everybody in this room. And I wish I could say, do this today. In terms of the viral-based therapies, there are risks. As I mentioned, with the adenovirus, it was 15 years or more ago, somebody died because there were safety signals that were kind of, we said, we're going to try this anyway. It looks like it might be okay. So there's a lot of caution when developing new things. It can be frustratingly slow, but as Mark Guillet and I will try to impart in the closing session here, we now have a number of different approaches that are being developed in the lab that have come through basic science studies, so we have a concept, and then a proof of concept showing that it actually works, and starting to move towards preclinical studies, so really creating something that could be tested in humans.
Are you using mouse models, or are you using the zebrafish models to actually do this kind of testing, or are you doing it in real tissue that comes from the repository?
All of the above, depending on the approach. So, for example, what Hank Granzier's lab does and what Jeff Widrick does is measuring how well the muscle contracts, and so they're finding chemicals that actually help improve the muscle contraction. So they're testing that in the muscle tissue that's been taken and it's been frozen, so the muscle cells aren't alive, but they're still able to contract, and so you can measure that and see the effects of the chemical. In the zebrafish, we can throw chemicals in the water or drugs in the water and see the effect on swimming right away. The mouse models take longer to work with, and this is one reason why things seem to are so frustratingly slow, because takes 30 days for the pregnant mouse to have a baby, then you have to wait 6 months for that mouse to grow up and see what effect it's going to have. But we're doing all of the above, and these are all either preclinical studies or proof of concept studies. Proof of concept is something where what you do proves that the therapy might work, but isn't in a form that can actually be used in humans. once you have that proof, then you can try to develop whatever your drug or therapeutic or virus is going to be, and then you test it. In terms of what you can do on a daily basis, sounds like you're doing all the right things. Physical therapy, and Mark can actually speak to this. Physical therapy, very important. OT, pulmonary, our plenary session this morning, critical to maintain your pulmonary function. Get your sleep studies, make sure that you're on optimal respiratory support, and then engage with the research community. So, yeah. Say one thing about any question.
Just one comment about how long it may seem to take things. I'm particularly interested in zebrafish models. So Alan said that the mouse 30 days when they're before you have your next generation of mice. With zebrafish, it's like a couple of days. So zebrafish, using zebrafish as a model, I think can really accelerate some of the questions we can ask and we can find answers to. Zebrafish are currently the second most if you look at the NIH grants that are funded, the top vertebrate model is mice, but the second model now is zebrafish. So it's not just in muscle. It's across biomedicine. But I think it's starting up in muscle now. I think you're going to see more and more zebrafish studies because you can get an answer quicker. It may not be the final answer, but it saves you time trying to get that same answer through mice. So you get an answer with a zebrafish, you can test that in a mouse instead of spending a year or two trying to answer that question in the mouse. So I think the zebrafish is one somewhat new thing in the field that could accelerate knowledge. And you want to talk about
Sure. And we're trying to use CRISPR-Cas9 to actually create these zebrafish models with ACTA mutations for several specific mutations so that we can test them for drug screens and small molecule screens.
Can I ask one more question? I mean, I can see on one side you've got the gene therapy track, and I can see that happening years and years away. And then you've got the other side of it, which is more focused on understanding how the muscle functions and how, what the commonalities are in terms of the dysfunction, so we can address that. But that gets addressed by drugs, doesn't it? As opposed, because you've got the genes over here, and then you've got the common dysfunctions. So is that, the 1,200 FDA-approved drugs that, hopefully one of them will stick? That's, sorry, but...
So they're hopefully going to be complementary. In myotubular myopathy, which is an example of a disease that is where we have a little bit further on in a few areas, there's developing gene therapy, but there's also an approach to alter the expression of other proteins that compensate. And that's going to start a clinical trial in a few months. And then there's another, tamoxifen, which some of you have heard of as a drug to treat breast cancer, does a number of different things in the cell. And what some of our colleagues have discovered is that tamoxifen also has a beneficial effect in that condition. So we could imagine in the hopefully near future, a situation where a baby is born extremely weak, they're too young and too weak to be subjected to gene therapy, and we may want to wait for gene therapy till somebody grows a little bit larger so that we can treat their whole body as a larger person. So then, for example, tamoxifen treatment might be initiated at birth, which is going to be nothing here is a cure, but it's going to be maybe the least effective treatment, but have a significant benefit. And then over time, then when that child is a couple years old, perhaps we'll do the gene therapy. One concern for the gene therapy is that that is likely to wear off over time. And because our immune system is going to respond to the gene therapy, it may not be as effective the second or third time we try it. So then in that disease, if the gene therapy wears off and somebody's 15, 20 years old, they need another treatment, perhaps it would be the so-called Dynamin treatment, which is this other approach to alter other proteins. So what we want to do is have an armamentarium of approaches. And this is what's been very effective in cancer. Many cancers are no longer a death sentence. In fact, people, often they're not cured completely, but they live 20, 30, 40 years longer with their condition. HIV or AIDS is no longer a fatal condition. When I was training, this was a death sentence. Now people live normal lives. They take a cocktail of pills. It's a variety of different things. So that's really, I think, what our goal here is. We're not going to be able to take a child with Nemaline myopathy and turn them into an Olympic track athlete. But there's a lot that we can do that will benefit them in between those extremes.
Well, thank you all for working on it, and thank you very much for answering my questions.
Sure, I hope that helps. You had a question here.
My question was just regarding, my son is 11, he has the, I don't know if I need this, he has the Nemaline gene. So my question was, is there going to be a possibility for gene therapy for that gene? Obviously it's enormous, and is there a virus that can do that?
So Nemaline in particular is so large that we're not thinking about gene therapy replacement approaches or viruses. But would you like to comment? You have some thoughts. This is world experts on Nemaline.
Thank you, Alan. Yes, so Nemaline is very difficult to work with, and we usually, the common methods that are used in gene therapy, they don't work for Nemaline. And what I have personally been working with before is we try to modify the RNA so that, for example, we could change one of the exons that carries the mutation. We could exchange that into a healthy one. But unfortunately, all families have their individual combinations of Nemaline mutations, basically. So that wouldn't be very effective possibly for a couple of patients, and to develop a therapy for 2 people, that would be very expensive. So, and what I think we've learned from Duchenne muscular dystrophy, it's probably not that effective, and we would have to use that all the time, because the RNA that we would target is replacing all the time. So it's, yeah, Nemaline is very difficult to work with, unfortunately, but we are constantly trying to find new methods to change, hopefully in the future, something that is wrong. Thank you.
All right, thank you, everyone. We'll have to wrap up this session. A warm thank you to all of our panelists who have joined us today to answer our questions.